All right, let's go ahead and get started. So I'm happy to open up this session where we're going to be focusing on open science data for space biology and health research. My name is Lauren Sanders. I'm the project scientist for the NASA Open Science Data Repository at NASA Ames. And I'm joined here by several of my colleagues who can help answer questions in the chat, post links in the chat, as I'm talking about the work that we've been doing over the past several years. So just kind of backing out for a moment, the open science work I'm going to be talking about today is motivated by the idea of traveling in space, space flight, and the upcoming deep space travel that's being planned by NASA. So in space flight, there are several environmental factors that are different from Earth that become very biologically relevant because they have effects on living systems. So for example, we can talk about altered gravity, elevated CO2, increased levels of radiation exposure, altered day and night cycles, so circadian rhythm changes, altered temperature and pressure, as well as the isolation and distance from Earth that become difficult over time. And so each of these factors needs to be taken into consideration and studied to make sure that we're engaging in space travel as safely as possible. So the NASA Biological and Physical Sciences Division is prioritizing this idea of not only traveling in deep space, but even being able to thrive in deep space. So this acronym TIDES. And so the idea is to study all of these deep space stressors that I just talked about by using a variety of platforms. So lunar platforms, free flyers, the International Space Station, ground-based experiments, and so on and so forth. And the overall goal is to study the fundamental mechanisms and the kind of genetic and physiological susceptibilities of biological organisms to this deep space environment. So the idea is kind of starting with these smaller or simpler organisms and then progressing up to larger, more complex organisms as the technological resources in space mature and we're able to study those organisms. And then alongside this science, we also want to be considering the technological advances, like what's needed for us to be able to sustain the long duration science experiments in deep space. So thinking about crop growth, habitats and ecosystems, among others. So specifically in the NASA space biology program, where most of the work I'm going to talk about today is taking place, we focus on characterizing the different risks to living systems that come with these different environmental exposures. So we leverage data from a variety of analyses and experiments, and I'm going to talk about how we're working to make these data publicly available, standardized, well curated, so that the scientific community at large can access them and reuse them. So specifically, NASA Space Biology investigates these 5 elements. So starting at the molecular level, all the way up through developmental processes, and then up through full organisms. And understanding these life processes at kind of the basic level contributes to both our fundamental knowledge here on Earth, but also has applications for human space travel. So what I'm going to be talking about today is our work at the NASA Open Science Data Repository, where our goal is to make all of this space biology data and any space relevant biological data reusable. Because the more people who have access to the data, the more knowledge we will gain to enable humans to thrive in deep space. So open data enables not just one research group to analyze a data set, but once that data set is public, many different researchers can analyze it and kind of amplify that knowledge gain. Then the more data we have available, the more advanced computational methods become available to us, because a lot of these recent computational methods like machine learning are very data hungry. We also provide access to environmental and scientific data from different spaceflight missions. So we can compare across missions, we can find disparities, we can find anomalies or similarities that can help us highlight these patterns across missions that may have taken place even years apart. And then this increases the value of each mission's data to be able to compare it with other missions. We can also compare across these different hazards that I was talking about, such as altered gravity, radiation. We compare across species or multimodal data types to kind of expand the complexity of these computational models. And of course, our analyses always benefit from longitudinal data to show the change across time. And then ultimately, with enough open space biology data, spaceflight exposure data, we're starting to see its utility for ecosystem platform development, countermeasure development, as well as the generation of food, pharmaceuticals in space, or the quantification of certain risks. And our final goal, of course, is to be able to involve the world in spaceflight, so we can make this exciting effort open to anyone who's interested to contribute. So with these goals in mind, NASA offers a number of biological open science resources. So I'm showing a few of these on the slide that I'm going to be highlighting throughout the rest of the talk. So the Biospecimen Sharing Program here on the left is a NASA internal program which is in charge of dissecting rodent tissues from flight and ground investigations and then preserving them, coordinating that internal tissue sharing and making sure that those tissues are preserved all in the same way and able to generate standardized data. Then the open source science programs include the NASA Biological Institutional Scientific Collection, or NBISC, and this is a collection of tissue specimens and microbial cultures which don't yet have data generated on them, but are available for request from investigators who have funding to generate data and study them and then make those data available back to the scientific community, hopefully. We also have data repositories, including the Ames Life Sciences Data Archive, which focuses on collection, curation of mission, project, and imaging data, and then Gene Lab, which focuses on collection and curation of omics data, so high throughput sequencing data. And over the past few years, NASA has made the decision to consolidate efforts and merge ALSDA, Gene Lab, and NBISC, these data and tissue repository I was just talking about, into a single one-stop shop data repository called the Open Science Data Repository. So this is making it easier for the scientific community, for investigators, for students to access spaceflight data across these different data repositories, and then also to see whether biospecimens might still be available from those investigations for request. So a few features of the Open Science Data Repository. We offer a single submission portal where investigators can come and submit their data so that it can be made publicly available and curated and standardized on the repository. We offer a user interface for research data submission agreements for investigators who have acquired grants and are ready to submit their data submission agreement to kind of show what data they'll be putting into the repository at the end of their investigation. Our data is all maximally open access, but we do have necessary controls for sensitive data, so I'll be talking about that in a minute. And all of our data is maximally fair, so findable, accessible, interoperable, and reusable. And we think of our Open Science Data Repository almost as an ecosystem. So I just talked about the data submission and the investigator workspace. We also have a data repository and multiple visualization platforms that I'm going to be talking about, which are enabled by the fact that we have a data processing team that works hard to standardize all of the data types into standard outputs that are reusable. We offer analytical bioinformatics tools to make the data as reusable as possible. We also offer training programs that I'll be talking about a little bit later, as well as community groups called the analysis working groups. And all of this goes together to enable new discoveries, publications through data reuse and meta-analysis, as well as generating novel hypotheses and grant submissions. So overall, just kind of looking at the data here from ALSDA and from Gene Lab. This is a breakdown of the different studies and data sets, species in our repository. One of the things that we're particularly proud of is the assay metadata table. So we collect the same standardized information from each experiment. We also have an in-house sequencing facility where NASA-funded investigators can have their samples sequenced. So I mentioned that we have several community groups we call analysis working groups. These are groups of users of the data in the database. They provide feedback to the database, but they also perform research analyses and publish papers together to amplify that knowledge gain. So the reason that these analysis working groups were originally started was because back in 2016, when GeneLab was getting off the ground and acquiring tons and tons of high throughput sequencing data at the terabyte data size, it kind of became clear that to make these data maximally reusable, it was going to be important to have consensus and standards for how to process these data through bioinformatic pipelines to create intermediate data products that are more accessible in terms of data size. Then when we realized it would also be very useful to have visualization and analytics tools to make the data access and reusability even higher, we continued working with the AWGs to provide feedback on those tools. And now, these days, what we've been working on is normalization, consensus, and data standards for larger meta-analyses or AIML analyses, which require lots of different data types to be merged together. So as of today, we have 6 analysis working groups. The animal, plant, and microbe groups focus on specific aspects of space biology. The multiomics and AIML groups focus on specific analytics of the data in OSDR. And the ALSDA working group provides feedback on the different types of data and making these metadata standards maximally reusable. So we invite everyone to join the AWG who's interested in contributing to space biology research. And one of the great things that we've seen come out of these analysis working groups is increased reuse of data and lots of new discoveries. So in this plot I'm showing on the left, what we see is that the original data provides a certain number of publications, but then kind of coming along right alongside it are the data-enabled publications. So publications that were enabled because that data was publicly available through the Open Science Data Repository. And a few of those latest publications are here on the right. So in terms of open data and analytics tools, I wanted to give a little overview of the types of data that we have available in the repository, some upcoming data, and then those analytics tools that make the data as accessible as possible. So in our repository, we have several data sets that come from both US-based, but also international studies. The European Space Agency, Japanese Space Agency, Canadian, lots of different contributors from international spaceflight biology. We have data from 45 different species. So this diagram shows a few of our different organisms, mainly rodents, because a lot of the research that NASA has done has focused on the rodent research missions, run research program. But we also have a lot of microbial data, cell line data, and different types of model organisms, which enable studying different physiological systems. We kind of break our data down into whether it's omics or non-omics, physiological, phenotypic data. So on the omics side, the majority of our data is transcription profiling, RNA sequencing, or microarray, but we do have quite a few data sets focused on DNA, a whole genome, methylation, metagenomics, and some proteomics as well. And then on the non-omics, so the physiological or phenotypic side, lots of different types of assays that study the impact of spaceflight on different levels. So from the molecular level all the way up through animal behavior or different ways of measuring the effects of spaceflight on proteomics or muscle. So putting all of this data together is really valuable for creating multi-level understanding of what's going on with a whole organism in space. Just kind of showing some of the exciting data that we're looking forward to collecting in the next few years. The European Space Agency is projecting out to collect up to 300 terabytes of omics data in the next 5 years. So most of this will be transcriptomics. And they've requested that all of their omics data be hosted via the Open Science Data Repository. And this formal agreement is currently in progress, but the idea is to centralize space omics data for the scientific community rather than having multiple siloed databases. So just one Open Science Data Repository, having all the data in one place will facilitate cross-analysis. So I'll show in a minute our multi-study visualization platform where you can pull in different data sets, and also avoids infrastructure duplication. So having all the data in one place rather than having multiple databases. We also very soon will be publishing data from the Inspiration4 mission from the investigator Chris Mason. So Inspiration4, for those unfamiliar with it, was the first orbital spaceflight with only private citizens. So Inspiration4, or we also call it I-4, had 4 civilian astronauts, flew to 364 miles in altitude and lasted for almost 3 days. So we have the Dr. Mason's group has submitted to OSDR 10 different data sets that were collected from these civilian astronauts either before, during, or after the Inspiration4 mission, which is exciting because this is kind of the first time that we'll be able to look at the effects of spaceflight on all these different human levels from these civilian data sets. We're also looking forward in the future to additional commercial or civilian human spaceflight data, for example, from the Galactic 5 mission. Now, with the advent of civilian spaceflight data into the Open Science Data Repository, we have implemented an access request process to keep data that needs to stay private until a request has been reviewed. So our process is inspired by the National Institute of Health dbGaP request process. So an investigator will submit a data request, which is then reviewed by a data access committee and either approved or denied. If it's approved, data will be provided to the investigator via a secure transfer. And at the closeout of the project, the data will need to be deleted. So just showing an example here of what it looks like on the data repository to request access to those restricted files and move into this request process. So we're operating on this idea that data should be as open as possible, but sometimes does need to be closed, but only as closed as necessary. Whereas metadata can almost always be open. So we can always see what data is available, what data can be requested, even though you do need to go through that request process. So with all of these new data types coming into our repository, we've been working on enhancing our search and data submission. So for those of you who haven't seen it before, this is what the front page of Open Science Data Repository looks like. You can either continue on to the data repository, search through the data sets, or if you're an investigator with data to submit, go through the submission portal. So in our data repository, we have the ability to search through multiple filters and search terms, including what data type you're interested in. So are you interested in a study, so a data set, or searching through different payloads that have been to space and kind of finding one that fits your research needs. You can also search through assay type, organism, tissue type, and filter that out. And then each of these records shows you an individual study. In the data submission portal, this has also been enhanced to include multiple different assays that an investigator might be incorporating into their experiment or their grant. So omics assays such as amplicon sequencing or these phenotypic behavioral physiologic assays, so non-omics assays like bone microstructure, calcium uptake, behavioral data. So as an investigator coming to submit data to the repository, you're able to select the assay that you're interested in based on the experiments that where you have data that you'd like to submit. Or if that assay is not available, you can work with our curation team to develop that metadata template so that investigators coming along after you will also be submitting the same standardized information for each experiment. So with that in mind, we also provide interactive visualization platforms, which allow users to mine through the data that we make available in an interactive environment. So I'm going to be highlighting 3 of those platforms today. The multi-study visualization app, which allows users to combine currently RNA sequencing studies that have processed data on the repository, merge those studies together, normalize them, and take a look at the visualization output. I'll also be talking about RadLab, which provides access to radiation sensor data from space, and the environmental data app, which provides access to both radiation data and other types of telemetry data, like temperature and CO2. So starting off with the multi-study visualization platform, this is a platform where you start with a metadata dashboard where you can kind of filter through all of the data sets that are available on the repository. Again, you can search through what's the experimental factor that was being studied in that experiment, what was the assay, with different organisms or tissue types. And once you've selected out your filters, you then are presented with a table of data sets that fit those filters. And you can select the data sets that you're interested to analyze and take forward into the visualization platform. So within the visualization platform, you're given a few options. So first, you have the option to normalize your RNA sequencing data sets using the DESeq2 median of ratios method. So this is a method which adjusts for differences across sequencing depth between samples. So this ensures that you're going to have an accurate comparison of gene expression levels across all of the data sets that you've combined. And this helps mitigate biases, and it enhances the reliability of the downstream analysis that you're going to see, such as differential expression analysis. When you're comparing between samples, it's important to normalize. You do have the option not to normalize if you have reason to believe that that's not going to be relevant for your study. Then on the next page, you're given the option to compare the results of your normalized and unnormalized data using PCA plots, so you can kind of see how the normalization adjusted the samples and see if there are any remaining biases that you need to take care of. And you can do that by selecting additional factors from the factor selection table in case you want to go back and normalize using a different factor. And then once you're happy with your normalization, you can move on to the third step, which is visualizing your data and analyzing the outcome. So we provide multiple different types of plots and analyses, including pair plot, volcano plot, and we also perform gene set enrichment analysis. What I'm not showing here is we also have the differential gene expression table and an ideogram. So check out our multi-study visualization platform. It's a useful way to be able to combine studies relatively quickly. We also provide the Environmental Data App, or EDA. And so this is another interactive visualization portal, but this one is focused on telemetry data and radiation data that's been collected from spaceflight missions. So here I'm going to be focusing on the telemetry data. So we provide readouts of temperature, relative humidity, and CO2 levels. And right now, we're focusing on those rodent research missions. So the mission dashboard is broken out into all of the different rodent research missions. And when you click on one of them, you get a time series plot of the time of that mission, time duration of that mission. You also have the ability to compare data from 2 different missions. So for example, here I'm showing temperature on the ISS and temperature on the ground for the time duration of Rodent Research 1 and Rodent Research 3. So you can compare those. And you can also toggle on and off the biologically relevant mission milestones. Like when did the mission launch? When were the animals transferred into the International Space Station? The different times that animals were sacrificed or returned to Earth. So this is a really powerful tool for being able to take the data that the biological data you're interested in from the repository and correlate it or compare it to what was going on environmentally on the ISS or for the ground control samples. And then finally, we've recently released RadLab. So this is a portal for providing access to radiation telemetry data from multiple databases maintained by multiple space agencies. So this is a real international effort. And we provide both a web interface and an underlying API for data retrieval at a programmatic level. So the web interface I'm showing here, you can see the time span of all available detector readings from these different detectors and locations. You can also select a specific instrument and spacecraft that you're interested in viewing and look at that time series. And you also have the ability to compare 2 detectors. We also deploy the ability to look at radiation levels in a geospatial format. So you can see as the ISS is traversing the Earth, the different levels of radiation based on detectors. So hopefully you'll be able to go and play around with some of these visualization platforms and find them useful to your research. Also in the spirit of open science, we have been developing training programs over the last few years, which both use the open data from the Open Science Data Repository, but also train the next generation of scientists in open science practices. So the first one I'm going to talk about is Gene Lab for Colleges and Universities, or GL4U. So this is a platform where we offer bioinformatics training that is specific to space biology. So the goals here are increasing accessibility and interpretability of our space biology data, training a diverse next generation of researchers, and enhancing awareness and understanding of these rare and precious space biology data sets. We organize our GL4U materials into 2 different types of modules. So we provide an introduction module, which kind of gives overview of space biology at NASA and kind of where Open Science Data Repository and Gene Lab fit into that ecosystem. And then we also provide a tutorial on how to use Jupyter Lab, because that's the platform that we use for teaching coding and analyzing data. And we give hands-on training in how to use the Unix and R programming languages. Then we dive into omics-specific modules. So whether you're in the RNA sequencing or amplicon sequencing, we've currently developed 2 different omics-specific modules for RNA-seq and amplicon-seq, where in these modules we go through explaining what's the experimental design of this particular omics data type, how do you properly process this data, and interpreting those results appropriately. And we do all of this in the context of hands-on analysis of an open science data repository data set. So data set that's already open that the students can go back to and compare to other data sets if they want to. We also offer direct and indirect training. So our direct approach is we train students. The other one is where we train educators. So then the educators are able to take those materials and run the boot camp themselves at their home institution. Our training is focused on minority serving institutions, MSIs, or historically black colleges and universities, HBCUs. We do provide compute resources to all the participants, and we make all of our materials publicly available on GitHub. And when we teach virtually, those boot camps are recorded and also uploaded and available on GitHub to enable independent learning. So I just wanted to highlight a few of the previously GL4U enabled workshops that we've done. So in 2021 and 2023, we taught direct student boot camps. So 2021 was RNA sequencing, 2023 was amplicon sequencing. And in 2022, we taught the indirect approach, where we taught educators, so 6 professors and 4 graduate students. And that was also focused on RNA sequencing. And those resources were made available to the principal, to the professors to be able to take and teach at their home institution. And one of those professors was able to teach this content to the students. And then finally, last year, GL4U partnered with the JPL Planetary Protection Center of Excellence. And this was providing space biology training in bioinformatics. And there were 2 workshop initiatives. So one was focused on the wet lab, which was JPL led, and then one was focused on the dry lab, which was led by Gene Lab. I also wanted to highlight an ongoing effort through NASA Transform to Open Science. So this is the development of TOPS, or Transform to Open Science, training materials called Science Core, where we're developing discipline-specific training materials that showcase open science workflows. So this was enabled through a grant on training in artificial intelligence and machine learning for space biological sciences and using NASA cloud-based data. So this is a training program that focuses now on artificial intelligence, machine learning training with a focus on space biology. So taking those machine learning methods that have been proved out in the larger biomedical field and bringing them in to focus on the open data that we have available in space biology, solving some of those tough problems that we have for characterizing the effects of spaceflight on living systems. We provide real-world use cases for the students. All of our hands-on training uses NASA open data sets, cloud-based scientific data sets, and I'll talk about a couple of those and how we develop those in the next couple of slides. But so what we're aiming for is to have this be a 2-week curriculum that can be taught in about 2.5 hours a day, or can be taken asynchronously or independently. And we're covering topics like introduction to space biology, how do you build a machine learning model, and how are the different idiosyncrasies of space biology data that you have to keep in mind for working with the types of data that we work with, how to interpret results appropriately. And then we have a few advanced models that we teach about, and at the end, the students are led through a capstone research project using open science data. So this program will be available in June 2024 for independent learning. And we're using platforms such as Google Colab and GitHub to ensure that students have access to the compute resources that they need to be able to complete the program. So stay tuned for that. We'll be excited to release that in just a couple of months here. So in the last part of my talk, I wanted to focus on some of the ways that we've seen in the past few years how access to open data and standardized data can enable computational advances. So some of the work that I'm going to be focused on here is from an initiative that we started at NASA Ames a couple of years ago called the Artificial Intelligence for Life in Space Working Group, or AI for LS. So the goal here was to see how we can leverage these advanced AI and ML computational methods to create models using the open space biology data from OSDR, so from Gene Lab and from ALSDA, and using these models to better understand the complex effects of spaceflight on living systems, but specifically across hierarchical biological levels. So pulling in and combining different types of data that help us paint that picture, not just on the molecular level, not just on the phenotypic level, but really pulling everything together to make a comprehensive model. So we use the open data from Open Science Data Repository, build models, and in the process, we also provide hypothesis generation and feedback back into the Open Science Data Repository. So in the AI for Less working group, we kind of think of machine learning as being able to touch multiple different parts of the space biology and space travel ecosystem. And so all the way from data standardization through to basic biological discovery, and all the way to thinking about how to improve biomonitoring and precision space health for long duration spaceflight. So one of the efforts that we undertook a couple of years ago to enable AI/ML efforts in space biology was through the NASA Science Mission Directorate. We published 2 standardized benchmark training datasets for space biology. So the goal here was, what are some common data types that are often found in space biology? How do we take open data and make it even more AI and ML ready? So we collected data sets from OSDR on RNA sequencing from mouse liver that had been flown in space. And one of the things that we were able to do here was a challenge we face in space biology is often our data sets are small. They don't have a lot of data points because the experiments are expensive or it's difficult, to fly experiments in space. So here we were actually able to use machine learning to make this data more AI ready by using a generative adversarial network to generate synthetic samples. And so we went from a data set with 112 samples to a data set with almost 7,000. And we showed that the synthetic data very closely mimics the real data, making it appropriate for training models. We also published a microscopy data set with fluorescent microscopy images of cells that had been stained for a marker of DNA damage and repair after exposure to radiation. So this is a great data set for studying the effects of increased radiation on the cellular level. So we converted the data set from its raw form into a maximum intensity projection that's a little bit easier to train models on. And we cleaned it up and published it with a few different labels that will make it straightforward for starting to train models. And both of these data sets are published on the AWS Open Data Registry. And these are some of the data that we're using for the capstone projects in the TOPS training. We also think about how machine learning can not only enable data standardization or enable data use, but can enable data management, especially as we're starting to collect more data in space that during spaceflight missions or long duration spaceflight that may not be easy to pull down to Earth, to downlink to Earth to do those analyses. And so here we wanted to highlight the idea of how can we use these advanced machine learning techniques for data management of these diverse data streams that are being collected in space. And we're excited to highlight a specific project, which was a NASA at Work project over the past couple of years called FLUID, which stands for Federated Learning Using In-Space Data. So the goal here was to train a global machine learning prediction model using 2 sources of data that never have to be in the same place. So one data set was hosted on the ground, and one data set was hosted on the International Space Station Spaceborne Computer 2, or SBC2. So this process leverages a technique called federated learning. So this is a methodology where a global model is trained by training models separately where the data is housed, then aggregating the weights of those models, but the data is kept private in the different locations and the weights are very small. So it's a lot easier to send the weights down to Earth to be aggregated. So the model that we're using in this particular implementation is a model that predicts causal relationships in data. So we've proved this out using 2 different data sets. One is a synthetic causal data set where there's known causal structure. And the other one is our radiation exposure cardiac data sets, which come from the Open Science Data Repository. And the cool thing about these data sets is they have measurements that conceivably could be collected on a future deep space mission, including ultrasound for a cardiac output or Doppler. All right, so we also think about machine learning for its impact on basic biological discovery. So we have the data. How can we create these multimodal models that tell us more about spaceflight effects? And here I just wanted to highlight two AI for Less projects that really show the power of these methodologies for generating insight into space biology. The first use case was a project that we did where we were trying to connect the effects of spaceflight on mouse liver through RNA sequencing of mouse liver from space-flown mice and a phenotypic measurement of liver disease. So this is actually a bioimaging measurement of showing that where in the liver are these fatty cells being retained. So we were identifying what genes in the RNA sequencing might have a causal relationship to this liver disease phenotype. So we were using a causal machine learning model called CRISP. And what we were able to show was, especially compared to more traditional bioinformatics or statistical methods, this machine learning method was able to identify several genes that had already been linked to this fatty liver disease phenotype. So that was really promising, and that paper is under review, but the preprint is available here. And then the second use case, we were wanting to use an explainable AI technique called Q-lattice, because explainability is so important in biology to understand where our conclusions are coming from. And so we were using Q-lattice to identify, again, bringing together different types of data. So we wanted to connect data that measured calcium uptake levels in muscles of mice that had been flown to space. So we know calcium uptake ability changes in space, and connect those with molecular biomarkers from different omics data types to see what at the molecular level can predict these calcium uptake aberrations. And the great thing about this explainable method was the results were interpretable mathematical methods. So they actually show the relationships between the biomarkers and then the relationships of those biomarkers to the final output, the calcium aberrations. So thinking about how machine learning can impact basic biological discovery, we've also been moving into how can machine learning, how can the new large language models and generalist AI impact basic biological discovery as well. So our goal here is to start with a large pre-training data set, create a generalized machine learning model that kind of understands the aspects of biology within that domain, and then refine it on small task-specific data sets. And what I'll point out here is all of this is dependent on having access to enough data and having that data be open and standardized and available. So in the project here, we trained a self-supervised model on a huge mouse RNA sequencing data set called Recount3, which is an open science data set, and it's been processed through the same pipeline so that all of the samples are comparable and standardized. And so we trained that generalized model to just understand what are those relationships within an RNA sequencing data set? What are those gene-gene network interactions? And then we refined that model on smaller data sets from the Open Science Data Repository. So, all right, can you classify spaceflight RNA sequencing samples from ground control RNA sequencing samples? And what we saw was that the generalist or transfer learning model far outperformed a model that was traditionally trained on the small data set. And so we're seeing a lot of promise for this approach, specifically in space biology, where we just don't have all that many data points to begin with. And finally, I want to end on kind of a more futuristic view of where open space biology and health data can take us and the processes that it can support for deep space exploration. So I want to highlight two recent studies that developed AI/ML vision and architecture to support NASA's deep space mission goals. So this vision was developed together with leaders in data science and machine learning from industry, academia, and government. And it's a decade level view. It's, what can we do in the next 10 years? It's very futuristic, but we were able to identify current machine learning technologies that could be focused on to enable autonomous systems for space biology experimentation and space health monitoring and support. And a large part of the discussion in both these papers focused on how open and fair data and data standardization can enable these technologies and processes. So, for example, one of the things that we highlight in these papers is a real-time proposed monitoring system that would unlock the ability to learn from these multiple streams of data across all aspects of, a spacecraft or space habitat and its inhabitants. So, starting with a continuous environmental sensing monitor layer, and including wearable sensors, and then thinking about even more invasive methods such as physiological monitoring. And so, together with methods like federated learning that I just talked about to avoid downlinking huge data sets, plus access to open space biology and health data on Earth, this kind of idea of multi-layered monitoring could enable real-time recommendations, especially in a situation where there are high latency communications, where it's very difficult to bring data to Earth or even to communicate. And then finally, ultimately, the kind of architecture that we proposed in these two papers would support a space health system for deep space missions. So bringing in diverse data streams from a variety of monitoring sources, being able to adapt to diverse extreme environments, and being able to provide real-time recommendations with human-in-the-loop iterations, with support from the crew and chief medical officer. But again, many of these recommendations were dependent on access to open and standardized space biology and health data on Earth to help pre-train these models, and then incorporating those same open science principles into the development of these more futuristic systems. So I'd like to conclude by thanking all the hard work from the Open Science for Life in Space teams and the Artificial Intelligence for Life in Space teams, and thank our support. And, of course, the Open Science Analysis Working Group members. And I'm happy to take any questions.
I have a question. Can you hear me?
I can hear you.
Oh, Ravi Sharma here. I have worked a little bit on space biology because I am an Apollo Achievement Award recipient during those days and during Skylab and Space Station. So I'm just wanting to know if you have found any modifications in genes when the astronauts return from long flights. Are there genetic changes other than just physiological?
That's a great question, and I think partly still an open question, but I will refer you to the twin study, which is one of the more long duration spaceflight studies that we have, and I can put that link in the chat. There are some interesting gene expression findings in that study.
There are I'm not only interested in finding the differences in the genes of the twins, which of course is a good subject to study, but also in the single subject of a single astronaut, what does duration and gene expression or modification imply? Do we have data on any changes in genes? And if so, then accordingly we can either modify future space medicines or target gene therapy for long duration space missions.
Yeah, this is a great question. And I think in the twin study, it is a single astronaut. So you may be able to find some good information in that study. Let me put the link in the chat for you.
Thank you.
Other questions? I think we're getting close to the end of our time.
If there's no other question, I want to ask if you found in your study again relating to genes, any particular aspects, like we do know that bone density is reduced, etc. So are there any mappings done on affected genes from any studies, on mammal studies or human studies?
Yes, absolutely. So I would now refer you to the data in the Open Science Data Repository. So a majority of our data sets are gene expression, RNA sequencing, and microarray. And you can use the multi-study visualization and analysis platform that I talked about to identify, to compare different samples from different exposures or spaceflight duration and see if there are any genes being affected. And it looks like my colleague just put the link to the twin study into the chat,
Yeah.
Garrett Backelman.
Yes. I see that. I will copy that certainly, or save the chat.
All right.
Therefore, I surmise that we can use your data to study the database of biological changes and gene changes, right?
Yes, that's right.
Thank you. Very good.
Thank you for the questions. All right, if nothing else, thank you all so much for being here. Have a wonderful rest of your day.